So as we know that the today topic is the porphyrin, and here we will study about the introduction to porphyrin. Uh, Porphyrin is a cyclic compound. A cyclic compound it means it's a compound that contains so many rings, okay, and they are interconnected rings. So we say it is a cyclic compound. Each porphyrin molecule, if you see the structure of porphyrin, so each porphyrin molecule, so this is a single porphyrin structure, okay. So each porphyrin molecule, there are four pyrrole rings are there. One, two, three, four pyrrole rings are there, and each pyrrole ring has four carbon and one nitrogen. You can see the pyrrole ring second number. Here also we can see four carbon atoms and one nitrogen. Four carbon atoms in the third and one nitrogen. So this is what the typical characteristics of the pyrrole ring and and the porphyrin molecule. Okay, porphyrin molecule always have a four pyrrole rings, and these pyrrole rings are connected by each other by methylene bridge. So here methylene bridge is this is the typical structure of methylene bridge. You can see in the structure of porphyrin here. So this is the methylene bridge, CH double bond and the single bond. So this methylene bridge or methylene bond, which is a alpha methylene bridge, alpha methylene bond, which connect pyrrole ring first and the second. Similarly, in the methylene bridge, beta type connect second and third pyrrole ring, and the gamma methylene bridge connect third and fourth pyrrole ring, and the delta methylene bridge connect fourth and the first pyrrole ring. So this is a cyclic compound, cyclic molecules where the pyrrole rings are connected to each other by one of the typical bridge, or what we say the bond, that is called as a methylene bridge or methylene linkage or methylene bond. Commonly, these reactions takes place, or these compounds are commonly found in here heme and the chlorophyll molecule. Heme is one of the pigment of blood. So heme is a hemoglobin component, and here chlorophyll is also the one of the pigment present in the plant. So if you talk about the heme or if you talk about the chlorophyll, the structure contain porphyrin molecule. Okay, and this porphyrin molecule contains this four specific pyrrole rings connected by methylene bridge. So we have understand the porphyrin molecule, the introduction first. Now coming to the question, that if there is a question in the exam to write what is the structure of porphyrin, so you can write in your own language, but here the answer is like porphyrins have complex cyclic structure because it's a cyclic compound, and structurally the porphyrin consists of four pyrrole rings linked to each other by which bridge? Which linkage? It's a methylene linkage. Pyrrole, if you talk about pyrrole is a five-membered closed structure containing one nitrogen and four carbon atoms. So here the pyrrole has a five-membered closed structure. Okay, that we have studied. There are four pyrrole rings are there. So each pyrrole rings, if you see the structure, it is a five-membered closed structure, which is made up of one nitrogen and four carbon atoms. So this is how you can explain the answer for what is the structure of porphyrin. How is porphyrin made? So here also it has a similar answer. Porphyrin is a large ring-like molecule which consists of four pyrrole rings, and which are smaller rings, okay, made from four carbon and one nitrogen. So pyrrole rings are what? They are the smaller rings, very smaller rings. And if you see the carbon atoms or nitrogen atoms, then you find that there are 4 carbon atoms in the each pyrrole ring and 1 nitrogen atoms in the each pyrrole ring. Okay. Now come to the heme. What is the heme actually? So as we are going, heme is a important biomolecule, important part of the hemoglobin, and this heme is a porphyrin molecule that we have studied in the introduction part. Okay, heme is a porphyrin molecule, and here it has a tetrapyrrole ring also. So as we know that porphyrin has a 4 pyrrole ring, so here also heme also has a 4 pyrrole ring. 4 tetrapyrrole means tetra means they indicate 4, and there are 4 pyrrole ring in the heme also. So the 4 pyrroline are interconnected by a methylene bridges. So this is the methylene bridges, alpha, beta, gamma, delta. These are the methylene bridges which connect each pyrrole ring. And the ring also contains the specific groups like vinyl groups, methyl groups, and propionyl groups. So you can see here that alpha, this is the the first pyrrole ring. The first pyrrole ring and the second pyrrole ring both have a methyl and vinyl group as its carbon, whereas pyrrole ring that is third and fourth has a methyl and propionyl group at its ring carbon atoms ring in the ring. So this is what we study the structure of the heme here, which is made up of porphyrin molecule. Okay, and here we have another important atoms, that is iron atoms in the center. Okay, it is kept in the center of the porphyrin ring. Okay, by interconnecting with the 4 nitrogen atoms. So this is what the 4 nitrogen atoms, each from one pyrrole. Okay, and they usually interact with the iron molecules. Okay, and together this entire structure is the structure of the heme here. Heme actually biosynthesis takes place in the liver and the erythroid cells of the bone marrow, and this structure, the structure of heme together with the iron and porphyrin, it gives red color to the blood. Okay, so the blood is red because of the heme structure. And what are the steps available to synthesize heme? So in our body, the heme synthesis is an enzymatic process, and there are so many steps are there. So step one is the first step, and in the first step there is a formation of delta amino labulinic acid. Okay, delta amino labulinic acid is the first product which are formed in the step one of the heme synthesis, and this step one starts with the precursors glycine and succinyl coenzyme A. So we need glycine and succinyl coenzyme A to make delta amino labulinic acid, and also we get carbon dioxide and coenzyme A as a byproduct. Okay, so here we have the product delta amino labulinic acid. Okay, and this delta amino labulinic acid, which is formed in the first step, now it is taken to the second step. So where 2 molecule of delta amino labulinic acid produces porphobilinogen. Okay, or porphyrinogen. Okay, porphobilinogen or porphyrinogen is a similar same things. Okay, so 2 molecules of delta amino labulinic acid makes porphyrinogen. Okay, and here also we get water also. Okay, in the third steps, what happens that there is a formation of porphyrinogen, which is made by 4 molecules of porphobilinogen. Here, this is the porphobilinogen. So porphobilinogen, if it is 4 in number, so it's 4 molecules of porphobilinogen combined together, and it makes a uroporphyrinogen third. Uroporphyrinogen third is made from 4 molecules of porphobilinogen. So in the step third, we have got uroporphyrinogen third here. In the fourth, we usually get fourth step, we can get the heme formation, So how it is formed? The uroporphyrin third is further converted to protoporphyrin 9, and we get 6 molecule of carbon dioxide as a byproduct. And here protoporphyrin 9 here combined with iron molecule, and we get a heme molecules here. So at last we have got heme when protoporphyrin 9 binds with iron, okay, and we get a heme. So we studied the steps of heme biosynthesis, which occurs in the bone marrow, basically erythroid cells, and also in the liver, which makes cytochrome proteins, which contain uh, heme there. So porphyrin has a function. What are the function of porphyrin? The porphyrin ring, as you know, that it's a ring, it's an important ring present in the heme molecule, and it serves as an important function. Okay, the nitrogen molecule of pyrrole ring. Okay, nitrogen molecule basically of the pyrrole ring, which are at the center of the ring. It holds an iron molecules. Okay, so this iron molecules yeah, is being uptaken by the porphyrin, and finally it makes the structure of the heme. So the porphyrin structure holding iron molecule gives blood its red color. Okay, so this is what the structure as well as the function we have studied for the porphyrin. And now we have come to the end of the slide. We have completed the lecture. Now, if you have any question, please ask me. I hope you have understand.